Hello, I'm Dr. Raajit, Chanana, medical oncologist, Dharamshila Narayana Hospital. So I'll be discussing regarding testing platforms and quality assessment in precision oncology. So to begin with, precision oncology is defined as molecular profiling of tumors to identify targetable alterations. So the basic goal is to deliver the right cancer treatment to the right patient at the right dose and at the right time. Goal of precision oncology has begun to be realized through multiplex molecular testing, including NGS. Oncologists should be familiar with technical aspects of NGS to facilitate selecting the most appropriate and cost-effective testing platforms. Considerations for molecular testing include which type of tissue to be utilized, timing of profiling in the disease course, the extent of panel to order, and degree of clinical annotations reported. Because of large number of driver mutations, actionable biomarkers, and targeted therapy, non-small cell lung cancer it has become the paradigm for precision oncology. The interpretation of molecular data to facilitate best practice still remains a challenge, and clinical trial participation and sharing of linked molecular and clinical data sets are strongly encouraged. So what is changing? From primarily looking at the disease origin in the body to understanding what drives the disease at the molecular level, from one size fits all medicine to treatment decision fitted to a patient's unique disease profile. The molecular in the precision oncology started in 1998 with the BCR-ABL translocation 922 rearrangement in CML patient that was successfully targeted by the small TKI imatinib, which ultimately led to its US FDA approval in 2001. Genomic instability is a cornerstone of cancer. Current molecular cancer classification divides detected mutations into driver mutations and passenger mutations. Driver mutations, they confer a growth advantage to cancer cells. They are positively selected in the cancer tissue microenvironment. They are causally involved in oncogenesis. A driver mutation may not be required for the maintenance of a cancer, but it must have been present at some point during the cancer's evolution. Whereas a passenger mutation, they do not confer the advantage of clonal growth. They do not contribute to the development of cancer. They don't have a functional consequence, but they are present in cancer genomes because they often occur during somatic cell division. So any cell that acquires a cancerous driver mutation already contains biologically inert somatic passenger mutations in its genome. Colorectal cancer as an example of mutational evolution associated with genomic instability. So you can see in this figure that from normal mucosa to colorectal carcinoma, APC is the major driver mutation. From colorectal carcinoma to tumor growth and metastasis, there are other driver mutations like VEGF and TWIST1 which helps in this process. Typically, a colorectal cancer tumor contains around 40 to 100 gene coding alterations, which includes around 5 to 15 driver mutations, some of which may be important for tumor initiation. Example, the APC in colon cancer, while others could play a role in tumor growth like VEGF or in metastasis like TWIST1. Now coming to the biomarker testing methods. So each of these methods can be done using FFPE samples created from either primary or metastatic tumor tissue, pleural fluid, or cytological samples. Basic methods are FISH, IHC, PCR, and NGS. In FISH test, fluorescent probes are used to label and detect specific regions on a gene. Samples are viewed under a microscope. In the IHC, antibodies are used to detect specific proteins expressed by the cells. The chemical reaction creates a color deposit on cells that bind the antibody. The cells may be identified under a microscope. In the PCR and RT-PCR, relatively large numbers of copies of DNA are produced from minute quantities of DNA or RNA source material. The NGS uses micro or nano technologies to run massively parallel sequencing. Conventional tests such as IHC and FISH, they are fundamental precision medicine tools still in daily practice. For example, the IHC can detect changes at the protein level. that result from various gene aberrations, most commonly gene amplifications, but also from specific DNA rearrangements or point mutations such as ALK mutation or IDH1/2 mutations in glioma. The use of IHC has extended to biomarkers of response to immuno-oncology such as PDL1 expression or mismatch repair status, which determine the eligibility for therapies that are based on anti-PD1, PDL1 agents. FISH has been the gold standard techniques for determining DNA rearrangements like HER2, ALK, ROS. IHC is a laboratory test, is a basic test that uses antibodies to detect specific proteins in cancer cells. If the cancer cells express the protein, then the antibody will attach to the cells. Chemicals are added so that if antibodies are attached, the cells will change color, and this can be seen in a microscope. FISH is a technique which is used to detect specific DNA sequences. A piece of DNA that is complementary to the target DNA sequence is labeled with a fluorescent tag. The tagged DNA is inserted into cells from the tumor being tested. If the target sequence is present in the tumor sample, the tagged DNA will attach to it. Fluorescence microscopy can then be used to confirm that the tagged DNA is bound to the DNA in the tumor sample. PCR produces relatively large numbers of copies of DNA from minute quantities of source DNA. The original DNA is denatured into single strands. The next step is anneal complementary single strands of DNA to form double strands. Then with the help of Taq polymerase, extension is done. Then ultimately exponential amplification happens as the preceding steps are repeated. RT-PCR is a method that qualitatively detects gene expression. With RT-PCR, an RNA sequence is converted to cDNA using reverse transcriptase, which is then amplified using PCR. Now, before discussing NGS, we'll discuss the Sanger sequencing. Sanger sequencing was developed by Frederick Sanger in 1977. So it's a special type of PCR that is called a chain termination PCR. The DNA sequence of interest is used as a template, and in this mixture of dNTPs and fluorescent labeled dideoxynucleotides is added along with DNA polymerase. The chain terminated oligonucleotide fragments are then separated based on size by capillary gel electrophoresis. Since DNA is negatively charged, it gets attracted to the positive end. Then ultimately using laser excitation, the sequencing is done by using sequencing machine. So using the conventional Sanger sequencing chain termination method, it would take around 2 years to identify the entire human genome. So now comes the NGS. NGS technologies have increasingly substituted for conventional techniques such as single gene testing and for targeted mutational platforms in routine molecular pathology. With conventional techniques, each biomarker must be pre-specified in order to be detected, and a purpose-made assay is required for each analyte. In NGS, it can enable the simultaneous analysis of a broad spectrum of genomic alterations, including mutations, copy number variations, translocations, fusions in multiple genes. So NGS is basically a more efficient, cost-effective, and most importantly, tissue-saving tumor analysis as compared to serial single biomarker analysis. So NGS systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Different machines are available and have been developed with various different technical details, but they share some common features. So the different machines that are available are 454 sequencer, the solid system, the ion proton system, and the Illumina system. Foundation one uses the Illumina system. Coming to the basic workflow of NGS, the first step is DNA library preparation. So that is done by sample extraction, DNA fragmentation, and the in vitro adapter is then amplified using PCR. So clonal amplification is done either by emulsion PCR or by bridge PCR. Then the third step is DNA sequencing. So sequencing is done by using different platforms like pyro sequencing that uses 454 sequencing, sequencing by ligation that uses solid platform, sequencing by synthesis that uses Solexa technology. The next step is alignment and analysis of data. The alignment of the assembled sequence is then compared with the reference genome, and this data is analyzed and the variants are compared with the available knowledge. There are various online tools that are available that can help to aid the clinical decision making. So there are various websites like My Cancer Genome, Jack's Clinical Knowledge Base, uh, the clinical interpretation of variants in cancer, oncology knowledge base, clinical genome. So the results can be checked in these websites and they can help in our decision making. So what are the benefits of NGS versus Sanger sequencing? NGS has a higher sensitivity to detect low frequency variants, faster turnaround time for high sample volumes, comprehensive genomic coverage, lower limit of detection, and the ability to sequence hundreds to thousands of genes or gene regions simultaneously. So the important question arrives that who should we test it and when should it be done? So many patients with metastatic disease may be good candidates for genomic testing at variable times in their clinical course. Patients with disease, with rare disease where there is no standard treatment options, they are candidates for early molecular testing. And certain disease like lung cancer, where a first line decision depends on multiple markers such as advanced non-small cell lung cancer, the use of a multiplex NGS panel at diagnosis becomes increasingly attractive, given the growing number of targetable genes. And when other disease states exhaust the evidence-based lines of standard therapy, then panel testing is often appropriate if the patient is fit enough to receive further treatment. And the important thing is when we should not use it. In patients of early stage patients where we have a definitive treatment option available, they do not require somatic gene panels. They will not have actionable alterations provided by NGS beyond what can be ascertained from standard histological evaluations such as by IHC or by FISH. Whether to use a new biopsy sample or whether it can be done with the archival tissue. So there are discordant genomic results between primary tumors and in the metastasis site, although this is highly disease site specific. For instance, RAS mutations in colorectal cancer, they are an early event in tumorigenesis, and reliable actionable information regarding use of anti-EGFR antibodies in the metastatic setting can be obtained from primary tumor itself. Conversely, estrogen receptor 1 mutations conferring resistance to aromatase inhibitors in breast cancer appear to occur as a consequence of exposure to aromatase inhibitors, and they are unlikely to be present in the primary tumor. Molecular evolution of the tumor has been documented in many cases. This is most apparent in patients receiving targeted therapy, whereby one of the mechanism of resistance is secondary mutations in the gene of interest. In these circumstances, repeat biopsies should be the preferred specimen. Current applications of NGS sorry, includes targeted gene panels. So it range from 20 to 30 genes. So they have a clinical implication. They are limited to a pre-specified group of genes. These are called targeted gene panels. They use hotspot regions with known actionable mutations. They have a faster turnaround time and they are more clinically relevant. The other are the whole exome sequencing, which focus on the coding regions of all the base pairs of the genome, and the whole genome sequencing that can involve the analysis of the entire tumor genome, including the intronic regions. Now, in this table, we'll give some examples of the commonly used protein and the methods to test it and the targetable drug that is available. We know that ALK can be tested by IHC and FISH, and there are certain targeted drugs that are available. Similarly, BRAF can be tested by IHC, PCR, DNA sequencing, and we have options of dabrafenib, tramitinib, vimorafenib, encorafenib. Other possible targets include EGFR, FLT3, IDH1, 2, MET. All these can be tested by one or more methods, and they all have a targeted drug available. Now there are plenty of options, NGS options to order. So it's important that we understand what test we need to order. So it depends on the choice of assay and design, the cost of the test, the tissue quality, the turnaround time, which laboratory you have chosen, the bioinformatic analysis, the clinical interpretation. Just to give you an idea, there are NGS assays for breast cancer panel, for BCR-ABL panel, for lung cancer. There are plenty of NGS panels available for lymphoma, for leukemia, for GIST, and for whole genomic sequencing. Foundation One offers testing. Foundation One has heme and sarcoma panel. It has a liquid panel that is approved by FDA. So this liquid panel detects up to more than 300 relevant cancer gene and also reports MSI and tumor mutational burden. So just requires around 8.5 ml of blood. And this is also approved by FDA in, I think, in this year only. And it has a turnaround time of 2 weeks. So just to give an idea that NGS is much beyond the targetable drugs. So there are studies that are going on. For example, in this slide, you can see that which will determine whether NGS will also help in developing particular drug and will also help in determining the response rate to particular drug. So the first cutout shows that a large-scale liquid biopsy database of advanced cancer patients, the Guardian Health launches real-world clinical genomic platform to accelerate precision oncology drug development. And the second cutout is the ANSWER trial that will enroll up to 1,000 patients and follow them for up to 1 year. And it will show the accuracy rate in predicting patients' response to drugs. So the evolution of medicine, in the past, it was intuition medicine. That is, it was only clinical based on signs and symptoms. In the present, it is evidence-based medicine based on studies, based on clinical trials. Yes, clinical signs and symptoms also forms an important part, but the future definitely lies in precision medicine. That is a specified set of treatment to a particular disease and to a particular patient. So apart from Foundation One, there are top 10 companies. It's a rapidly growing market, precision medicine software market. There are plenty of other companies also. So just to give you an idea, they are To Be Precise, Peer and X, Gene 42, Translational, Fabric, Sophia. So in this slide, you can see that precision medicine is a growing industry with a possible $2.8 billion market by 2027. With a growth rate of around 11.8% per year. So we are so close, we are yet so far. What does it mean? That precision medicine has brought new complexity and challenges. So this is a slide from JCO 2011 paper. We have all seen it. Figure 1 is before initiation of vemurafenib. After 15 weeks, we have seen that it's an excellent response. All the lesions have almost disappeared, but the patient has relapsed after 23 weeks of therapy. So there is still a long way to go before we fully understand and fully developed the NGS platform and how to interpret the results of NGS. So with this, I would like to conclude and I would thank you for listening.
So that was a very informative and elaborate discussion on various tests we have. So few questions to bother you and difficult for all of us, but still. So how do you interpret results if you have different results in different platforms? So we have IHC.
Yeah, thank you, Dr. Chandrakanth and Dr. Parikh, sir. I'm not sure why my slides were blurred. So, but I'll take up the question. So I recently found a patient who was strongly ROS1 positive by IHC and it was negative by FISH. So in these ALK, I think if we get a positive test by IHC, then we usually don't go for fish because IHC has also become FDA approved. So if the test has also been done by fish and if it turns out to be negative, then I would like to test it by some other platform. But one thing is that these sets of patient would be chemo sensitive, so I would start them on chemotherapy and I would wait for the NGS test to come. But in patients that I had was ROS1. ROS1 only platform that is approved is fish. So if I was planning to get entrectinib for that patient, but the fish turned out to be negative. So these sets of patients, they are also chemo sensitive, and I have asked to get a ROS test by another platform like NGS.
So when we have a discrepancy, I think we need to have a deciding test and then decide further. Okay, next question.
Next question, please.
So as you correctly said, there is so much in the market to choose what to choose. So similarly, we have so many immunotherapies, so many targeted therapies. Now we have so many platforms. So how do you choose which one to use?
First, I would like to answer when to choose. So it is not an easy answer, but we should understand that those patients whom we run out of options, or those patients in whom the molecular mutations like lung cancer, they drive the treatment, that is the time when we should do order the NGS for the patient. And regarding as far as liquid biopsy is concerned, we know that it is not the concordance rate is around 60 to 80% as compared to a solid tumor. But as far as the choice is concerned, then it would be Foundation One since it is FDA approved. I'm not sure about Guardian 360 whether it's FDA approved or not. So my choice would be Foundation One, and definitely we have Indian labs also, which are offering the extended mutational test, especially targeted gene panel and whole genomic sequencing. So we also have plenty of experience with this lab. So best test would be to do the targeted panel, because we know that for that set of disease we will be able to find out those sets of mutation and we'll be able to get the answer in an earlier time. But if we are short of options, then we would like to do one entire home genomic sequencing. It can be done in Indian labs that are offering these. These are MedGenome, the Onquest lab, the Lalpath lab. So it's based on the comfort and whether they have validated their test or not. The technicalities of the validation you need to discuss with the lab itself.
Raajit, can I ask you one question?
Yes, sir.
All the labs, when they come to us, they always say we are the best, right? So if the same or similar test is being offered by 3 or 4 labs, how do you decide whom to send to?
Sir, the answer lies with you, sir. So based on the experience of our professors and teachers and what we have learned, so what everyone is comfortable with and what we are comfortable with, based on the reports, what report they provide, whether they are mentioning all the genes that they are testing or not, and whether they are confident enough to provide the data to support their test.
Right.
So all these factors are taken into consideration before asking the patient to go to that lab. So it's a very practical point,
right,
and
right.
I'm not sure regarding the theoretical aspect.
So Anuradha Chogle, if she's around, can we unmute her? Maybe she can throw some light on quality control.
Ma'am, you can unmute your mic. Anuradha, ma'am, you have to unmute your mic. Ma'am, are you there?
She just posted on the chat, so I thought she was there. She said Guardian 360.
I'm there.
Yeah, go ahead. Please go ahead.
Yes, regarding the, I agree, we have to send it to the best lab, but when you say, when you're sending it to the best lab, you have to see, from my perspective, it has to be NABL certified or CAP certified laboratory.
Okay.
Then only you can get the assurance of a good quality. And there are various steps in good quality control which are measured by NABL and CAP when you do the certification from them.
Okay, so I think that is something that at least objectively we can look at a parameter that do they have a certificate from NABL or CAP.
Yes.
Maybe if they are participating in an EQUAS program also, that would be useful,
That
right?
is also enough. Proficiency testing is also enough. Participation.